Hi everyone, this is Teng-Jui Lin. You can call me Owen. I joined the lab last November and today I am presenting on quantifying morphological heterogeneity of cells and nuclei with vampire analysis. This study was published on Nature Protocol this year by Wu and Wirtz. Traditionally, geometric parameters such as solidity, aspect ratio, and shape factors are used to describe cell morphology. However, they cannot represent morphological variability. For example, if we plot those parameters onto a 3D plot and find close points on the clusters, we can see that the shape of those clusters, even though they have similar parameters, have different shapes. In addition, if we add more and more parameters into the model can be hard to interpret. And this is where vampire comes in. Vampire, or visually aided morpho-phenotyping image recognition, defines morphological shape visually using shape modes. It also evaluates morphological heterogeneity using Shannon entropy during the analysis. Vampire analysis takes in the user's image that has segmented cells. The algorithm extracts the cell contour and finds n equidistant points on the cell contour. It then normalizes and aligns the cell shapes. Then it uses principal component analysis, or PCA, to reduce the dimensionality of the data. Originally, we have n points, and then those n points are projected to n principal components. The algorithm then selects the top principal components that captures 95% of the cells of the data's variability and reconstruct using those PCs to the cell shape. Vampire analysis then use k-means clustering to identify the shape mode. Each point on the graph represents a cell shape. The algorithm identifies clusters in the data points and then uses the centroid as the average representation of the shape mode. Vampire can be applied to analyze mouse embryonic fibroblasts. The cells are cultured on micropattern plates with triangles and circular shape and control with no pattern. The raw images are segmented into segmented cells and the algorithm identifies the cellular shape modes as shown on the right. On the left table, it shows the frequency of cells being in each shape mode. As we can see, the triangle pattern cells have more of a triangle shape mode in mode 1 and 2. The circular shape pattern cells have more of a circular shape in 3, 4, and 5. And the control with no pattern has a variety of shape patterns in all the shape modes. And these cellular variability of shape can be captured by Shannon entropy shown on the right. Whereas the circular shape cells have lower entropy, the triangle shape and the control cells have higher entropy. The geometric parameters of each population are similar, so it is hard to get information about the shape variability from geometric parameters. Vampire has its limitations. It assumes proper segmentation of the image and cells, and it applies to 2D images only. It also has limited spatial resolution since it uses principal component analysis to reduce higher dimension data to its lower dimension approximation. It needs large and expensive data set to get a good prediction on dominant subtypes, but it may have poor prediction on rare subtypes. So in conclusion, Vampire visually defines morphological shapes and evaluates morphological heterogeneity. As for the future, we aim to change the image inputs to individual cell inputs since we have limited sample size of mouse brain slices. And that's all for this presentation. Thank you all for your time.